So I will present you the development of SOM 2355 in Huntington's disease. SOM Biotech is working with its own artificial intelligence software to screen among known drug potential candidates to be proposed in new target indication in rare disease. With this method, Biotech, which is an antihypertensive drug used for years with no psychiatric problem, was identified as a potential and potent VMAT2 inhibitor. SOM is now currently developing this candidate, which was renamed SOM 3355. At the preclinical stage, the compound was tested in vitro, and its functional inhibitory activity at VMAT2 was confirmed. And then with in vivo studies, we have confirmed its brain penetration in sufficient amounts. So this led us to go very quickly in the clinical stage, and we have performed a first proof of concept study in Huntington's disease patient, and which has confirmed that SOM 2355 was reducing chorea symptoms in those patients. So now we will continue the development with a phase 2b, which will test 2 doses of SOM 2355 in a 12-week double-blind placebo-controlled study. Initially, Biotech was first developed in the 80s by Werner Lambert as a selective beta blockant to treat hypertension and angina pectoris. Currently, the drug is commercialized by Nippon Chemical in Japan and in China, with the Calvin brand name in tablets in the indication of hypertension at dose up to 200 milligram BCD, but also the 300 milligram BCD dose was tested in a clinical trial, and for up to 6 months it was well tolerated. So then this drug has an unchanged safety profile after more than 30 years of use in hypertension, and with a cumulative exposure of more than 2.5 million persons. And in the pharmacovigilance, we didn't find in the different pseur any concern regarding the CNS side effect, and particularly no issue regarding depression and suicidality. In vitro assays have confirmed the functional VMAT2 inhibition of SOM 3355 at the same potencies as tetrabenazine, but it differs by its binding to the tetrabenazine site on the VMAT2, and probably it is acting on it is interacting on a different site or in a different way to this site. Also, in addition, we have found that SOM 2355 inhibits the VMAT1 in the brain, which is a particularity as tetrabenazine does not has this potency. So this led us to make the hypothesis that SOM 2355 induce changes in dopamine signaling and improves motor symptoms, but it may preserve from psychiatric side effect, and it will maintain the mental health. Then at the clinical stage, we have performed this first proof of concept study in 32 Huntington's disease patients, and it was a double-blind randomized crossover study testing SOM 2355 compared to placebo in a crossover design. The primary endpoint, primary efficacy endpoint was the improvement of at least 2 points in the total maximal chorea score in any active drug, in any active drug period compared to placebo for each patient. And we have also a secondary endpoint, which were the clinical global impression of change, the patient global impression of change, EHDN2021 functional and motor subscales, and the safety assessment, and also with particular attention to the suicidality with the Columbia Suicide Rating Scale that was performed at the beginning and at the end. Other exploratory endpoints were the prolactin level in plasma and the plasma concentration of the drug. The result on the chorea an improvement of the TMC score of at least 2 points compared to placebo in any active period drug period was seen in 57% of patients. And at the highest dose, 200 milligram BCD, induced a significant improvement of the TMC score compared to the placebo in a mixed model. So those results provide a confirmation of the expected effect of the drug on chorea symptoms, and this effect is related to its VMAT2 inhibition. In addition, the clinical global impression of change and patient global impression of change were corroborating with those results, showing an improvement in at least 70% of patients when they were under active drug. Regarding the safety, so the safety profile of Biotech was unchanged in this normotensive population of patients with Huntington disease, and all the adverse events were mild or moderate, and the most frequent were bradycardia or hypotension, which are known side effects with beta blocker. And particularly no adverse event of depression or suicidality were related to the study drug administration. For the vital signs, we have seen a slight reduction of the mean heart rate and the mean systolic blood pressure during the period under the active drug, but still in normal range. And this is corresponding to the known cardiac effect of Biotech as beta blocker. The plasma level were mildly increased, not proportionally, with SOM 2355, and this is probably reflecting the effect on dopamine pathway by the VMAT2 inhibition. So for us, it was also confirmation. And for the PK data were consistent with the previous published value, and we have a dose linearity and proportionality. And then we made the PK/PD modeling, and assuming this dose proportionality, we can predict that if we increase the dose to 300 milligram BCD, we can expect a mean decrease of the TMC score of more than 2.5 points in the next trial. This is only prediction, but this corresponds to the same magnitude of the treatment effect of tetrabenazine. So the next trial will be a phase 2b, and it will be an international multicentric double-blind placebo-controlled study in parallel arm, which will test 2 doses of SOM 2355, 200 milligram BCD and 300 milligram BCD in comparison to placebo. We must include at least 129 patients with Huntington disease suffering from this choreic movement, and the study duration will be 12 weeks with 7 visits. The drug will be taken in oral capsule twice per day, starting with an up titration, and the patient will be allocated in a randomly manner to the 3 arms to receive either placebo or the active treatment at 200 milligram BCD or the active treatment at 300 milligram BCD. The objective of the trial are to assess the efficacy in reducing chorea in those patients and the safety of the two doses compared to placebo. The inclusion criteria, so we will recruit adult patients with a diagnosis of Huntington disease with a choric movement and with a total maximal chorea score of at least 10 points. Those patients should be able to work independently and have a total functional capacity score of at least 7 points, corresponding to mildly to moderately impaired patients. Regarding the exclusion criteria, we will exclude the patient with a juvenile form of Huntington disease starting with a onset before the age of 18. We exclude the patient with rigid akinesia. And we also formally exclude the patient who are taking tetrabenazine within the 3 months before the enrollment, or any other antichoric treatment within 1 month before the enrollment. An history of severe depression or suicide attempt in the last year will be also an exclusion reason. Severe untreated depression or active suicidality will be excluded. Also, patient can be treated with some authorized antidepressant and stabilized, but in certain conditions that we will see later. We will exclude also patients with hypotension, bradycardia, or orthostatic hypotension due to the nature of the drug, and the patient with hypertension already treated with at least with more than 2 antihypertensive drugs because of the combination. Any other active clinically significant illness will be excluded, and particularly according to the judgment of the investigators, or any abnormality that we can find in the ECG or the lab test. Regarding the concomitant medications, so of course, all the other antichoric treatment will be prohibited during the study, particularly tetrabenazine or derivate, but also neuroleptics, and all other drug with an antagonism acting on the dopamine pathway. However, some treatment will be authorized, and particularly some antidepressants, except the monoamine oxidase inhibitor. All the other antidepressants, particularly the SSRI, will be authorized if they are given at stable dose during the whole study. Some atypical neuroleptic may be authorized, but only if they are needed and prescribed for an aggressive behavior, not as an antichoric treatment. The other antihypertensive drug, if the patient has hypertension, other antihypertensive drug can be combined with bevonterol, but we exclude, of course, the beta blockers. All those drugs authorized will be given at stable dose during the trial. The study endpoint, so the primary endpoint will be the change in the total maximal chorea score between the baseline and the end of the treatment. We will have also key secondary endpoints, particularly the clinical global impression of change and many other endpoints. For the safety assessment, we will have a particular attention on all the adverse events and also on the cardiovascular parameters, and the assessment of depression and suicidality that will be monitored with a BDI, Big Depression Inventory, and Columbia Suicide Rating Scale. We will assess also the sleepiness, the akathisia, behavioral symptom, and cognitive disturbance, and the total functionality capacity, and the functional assessment will be checked at the beginning and at the end of the treatment for the safety. PK/PD assessment will be also performed. So an open label extension study for the patient having completed this double-blind study will be proposed to assess, will be proposed to those patients to assess the long-term use of the drug, and with a personalized dosing and visit every 6 months. So finally, this phase 2 protocol was shared with the EHDN and the annual clinical trial committee for review and advice, and we are expecting their feedback before finalizing this protocol. Approximately 20 sites in Europe will be invited to participate to the study, and they will be asked to include at least 6 patients per site. The site selection is ongoing, and it is still open. And the study is expected to start by the end of this year. So finally, if you are interested to participate in this trial, feel free to contact us. We will be very happy to give you more information. Thank you for your attention.